I have a lot of stuff to cover, so we're gonna have to go really quick. I was teasing with some people earlier that normally they give me half an hour to give my talk, and I feel it's not enough. They gave me 1 hour this time, and it's still not enough. So we'll see how it goes. Yeah. Okay. Before things start, I want to just make sure that I do a highlight of our NDSU Extension Bulletin A64. Those are the dry bean variety trials that we conduct around all the state, including Minnesota as well. So you're going to have those results there. All you have to do simply is just Google NDSU variety trials. Otherwise, you have the link there. All the information from our trials that you got to see and visit is going to be there. Okay. So when I say that one of my varieties is great, just don't believe me. Just go and check on the booklet and see if I'm lying or not. Okay. Okay. This is pretty much what we're going to cover today. I got 2 new varieties for you. I'm getting spoiled you a little bit because every time I come, I'm just talking about a new 1 or 2 varieties. We're going to talk about beans that pop, if you never heard about that. A potentially new but very mild disease we discovered this last season. I'm also going to tell you a little bit about tile drainage and the relationship with zinc nutrition and the things that we're doing there. Looking for the next rhizobium superstar, okay? The work that we continue doing with SCN, soybean cyst nematode, and a little bit of the modern tools that we're trying to use in the program to make our lives easier or better, I guess, or at least improve efficiency, right? Okay, so let's get into topic. Allow me to introduce you ND Red Velvet. It's a small red. After a lot of testing that we went through and did, we found out that it was a good option for the small red market that we have. We're not huge in small reds in the region, but still there is a significant portion of our acres that are being grown with small reds. Huge interest in Central America, so some of you are probably exporting to Central America because it's, in countries like Costa Rica or Honduras or Nicaragua, this is the main market class, right? With small reds, there are like 2 types. So the U.S. market and Mexico prefers larger size seed, but the Central American market prefers smaller seed, right? And it's because, they're just different in that way. This is more like for the North American and Mexican market. So this is a larger seed type, more comparable to Merlot, okay? Which has been a variety that has been around forever, right? And now we're able to have something else. So you have a comparison there with the usual suspects that are commonly grown in our region. USDA Lava is a new variety coming from the USDA program in Prosser, Washington. Viper has been around for several years, coming from the Provida group. They're here. And Merlot came out of Michigan State, 20 years ago. I mean, it's been a long time for this. So it's good that we have something to replace. Merlot, canners love Merlot, by the way. And people who process small reds, Merlot is kind of the standard, right? So I think we're getting close to that same ability for processing and canning. We'll see. Here is the seed size that I was telling you about, right? 33 grams per 100 seed, which is very comparable to Merlot, as you can see there, 33 as well. While the other 2 tend to be a little bit smaller in seed size. Again, probably more suited for the Central American market rather than for the North American market. When it comes down to yield, nothing really too crazy. It's competitive with everything else. We found out that it yields better than Merlot for sure. That's on your left side. And that difference is actually significant. But when it comes down to the other ones, there are numerical differences, but they're not statistically different, right, across all the trials and environments that we tested. So now you have something that it's pretty much good quality, larger seed size, upright plant architecture, and another option for the small red bean market. Allow me to introduce you the second variety. We call it ND Pink Slipper. And for those of you, this is a light red kidney, by the way. For those of you who don't know, Lady Slipper is the Minnesota state flower. Okay, so we try to pay homage to, the state of Minnesota, because most of the work that we do for kidney beans it's mostly thinking about our Minnesota growers, right? We don't grow a lot of kidneys in North Dakota. Most of the kidneys happen to be grown in the sandy areas of central Minnesota and all that area here. A lot of that process happen in farmers' fields that we have. We have some of them in Bobby's here. He's helping us. So, and many times during the development of this variety, we were doing selections and work at his farm as well. Here is a comparison of the seed size and shape. I think we are keeping the standards that are expected for a light red kidney and things like that. But we were trying to find or come up with something that were closer in maturity to what all the other light red kidney varieties are. In other words, we released Rosie several years ago. Rosie is a fantastic variety, but it's extremely late. And for some farmers, that doesn't work in what they do because they do both dark and light red kidneys. Usually farmers like to be done with light red kidneys so they can move to the darks. And this variety has been very successful just for those cases in which some companies are contracting just for farmers to grow only light red kidneys. So Rosie is really good, but it doesn't fit everybody else. So we were like, okay, we need to tune down the maturity to something that will be more comparable to everything else. And that's what you have there in that chart, right? Now we have that ND pink sleeper. When it comes down to days to maturity, it's on par with everything else that is being grown across the region, pretty much. There is a trade-off, and everybody knows this. Every time you're gonna have earliness, it's gonna be at the expense of seed yield. That's the reality of things. You cannot have both things, right? It's always the trade-off that happens in breeding. So every time you are reducing the cycle, you are also reducing the chances of getting higher yields. But again, we thought that this was what the industry and the farmers wanted, to have something that fits better into their logistics and their timeline in the farming operations. So here they are, okay? Same thing. I mean, I think in terms of seed shape, quality, size, all those things, we are meeting all the standards. I still personally prefer the color that Rosie gives you. You can see that it's more pink, it's more bright than everything else. And that's why some companies and some farmers really like it, in spite of the late maturity, but it pays off with yield as well, okay? I don't have time to talk about all the other varieties, but we have a booth somewhere here. I think it's 425, right? Oh, it's over there. They're pointing me. So it's on that side over there. Joycelyn and Juliana will be there. If you want to ask about seeds of any of the NDSU varieties, just talk to them and they will set you up. Even if you just want to try something and get a small sample or something, they will be able to help you with that. We have about 12, 15 varieties available. They're all for you, right? This is the work that is being paid with all the work that we do. There are more varieties. I would say Galaxy and Rodeo are probably the newest, and they are killing it. I would say I can almost bet a beer with any of you that ND Rodeo is beating any other slow darkening right now in the region in terms of yield, okay? So we're pretty excited. We think that Galaxy is gonna be a good replacement for Eclipse, which is kind of aging and getting old, right? Eclipse was released in 2005. It's been 20 years since the last time that Eclipse was released. So I think we need something that is going to be able to compete in that same scale and level. All right, let's talk about a new thing that we found this last growing season. You know, in late June, mid-July, we were looking at some of our fields. Actually, an agronomist from Carrington, called me one day and say, I think you have anthracnose in your field, in your nursery. And I'm like, oh, shoot. You know, it's very rare. I only have seen anthracnose once in my nursery. So this will be the second time. So we rushed next day to see what was going on. And we found it, but it was very mild. I mean, you really need to know what you're looking for in order to find the anthracnose there. It was stronger in the kidneys and cranberries later down in the season in Minnesota, but nothing that would be too crazy. For me, the most striking thing is I know that some of the varieties that we had in those trials are resistant to anthracnose, and they were showing symptoms. And that's when you start scratching your head, right? It's like, what's going on here? This guy is supposed to be resistant. There's something going on here. Michigan, I call the Michigan guys because they get anthracnose more than we do here. For them, it's a very common disease. Uh, and they say, yeah, we also notice that there is a weird type of anthracnose that it's not your typical anthracnose, but it looks like, but it's not. And then we're all confused trying to figure out what to do, right? So of course, we also notice symptoms in soybeans and some other weeds that are probably acting as alternative hosts for the pathogen, right? So we took a bunch of samples and sent that to the lab along with the plant pathology group, you know, Dr. Malaika Eber and all these people to figure out what's going on. But this is pretty much how it looks, right? And my goal with this is not to scare the hell out of you. It's just to show you so you can have your eyes open when you're in your fields walking. Look at these are on the upper side, you have the symptoms from the dry beans. On the lower side, you see the symptoms on soybeans. So soybeans also get affected by this somehow, right? Which is concerning because we grow a lot of soybeans too, right? The intraveinal necrosis is a very typical symptom of anthracnose, right? Bean anthracnose, and also those spots that you see in the pods, right? Those are the worst cases. Don't think that it was always like this. It was much more milder. I'm just showing you the really, really bad cases here, right? Those little stains on the stem are also very common. Anyway, I'm just putting this in your radar, and I think I did that during the dry bean field days, not with the objective of scaring everybody about a potentially new disease, but more as to keep your eyes open. We're working on it. Right now, we think that there are 3 potential new species that could be causing this, right? So your common bean anthracnose is called by a fungus known as Coletotricum lidemontianum. We're finding, based on different things that we're doing in the lab, and at the DNA level, that this might be a different species of Coletotricum, right? And there are 3 potential candidates. I don't want to venture myself. We have a pretty good idea that might be one of them, but I don't want to venture to say which one it is yet until I'm 100% sure, right? I'm a scientist, remember. And some of those have been previously reported, at least on soybeans and dry beans. So we're going to continue working on this. We're also trying to screen all the common cultivars that we have in the region with this in the greenhouse, just to see if by chance any variety is at least tolerant to this, right? We don't know yet. We noticed that one of our black bean breeding lines was totally immune to this. So at least there is hope that could be some genetics that is going to help us. I didn't see anything on our current commercial varieties that we all use that I could call as resistant. So more to be seen. I'll give you an update next year about this, but just wanted to put it in your radar, okay? How many of you have ever heard about Nunez? 1, 2, 2 people in the room, right? So for those of you who don't know about this, Nunez are beans that came from Peru. So the Incas actually were the ones kind of domesticating this type of bean. It's a bean, right? And what they happen is, as you remember, Incas are in the high altitudes in the Andes. It takes forever to cook beans, right? I mean, the water boils, but it takes longer to cook those beans. So being so resourceful as humans you really are, they say, well, why don't we roast them instead of cooking them? And therefore, they realize that some of these beans actually had the ability to pop, not in the same way that a popcorn will do it, but they kind of swell more like a peanut, think about, right? Something like this. So they're gonna look something like this, right? So you have popcorn. Why popcorn is popcorn? Because it has modified starch, and also the pressurized steam that happens is what creates the seed to explode. In the case of popping beans or Nunez, they only are doing that because of the pressurized steam. So the starch in the seed is the same, pretty much, right? So those are kind of the main differences. So as you can see, they never pop as what you see in a popcorn, but they get really, really big. And then after that, you can just go and snack on them. My crew in my lab is crazy about these things. They're constantly, every time I show up in my lab, they're snacking on these things. So they love it, yeah? Because you put it just 2 minutes in the microwave and you can eat them, same as popcorn. Okay, so the issue with Nunez is that they are what we call photoperiod sensitive. In other words, we have long days in our growing season, right? 16-hour day length. these things don't flower. They stay green, 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 and never flower, right? Because this is not their place. They're adapted to the highlands in Peru, but not here. So breeders have been able to start making crosses and trying to make that material adapted to our conditions, right? So that's why we're trying to make the differentiation between what is a Nuña bean, which comes from Peru, and what a popping bean is, which it was developed through breeding, right? Just also to avoid cultural shock and things like that. So that's pretty much the story I wanted to say there. Luckily, we got a project funded by USDA last year that is going to having a lot of people, institutions involved into trying to promote this and go forward with this. Dr. David Gang, I know he's in the room. So if you want to talk about popping beans, he's your guy, right? He's our project director. I'm also involved into this, but as you can see, there is also other universities that are doing this. David is a professor at Washington State University, and that's our lead institution for doing this. And in part, it's because for those of you who don't know, the germplasm collection that we have for beans, the largest collection of beans in the US, it's in Washington State, right, in Pullman. We have about, for Nuñas at least, we have a collection of about 80, 84 accessions, and we've been growing them and using them to do all the work that we're doing in this project as well. So these are pretty much the objectives that we have in that project, right? We're going to enhance whatever material we have. Some of them are very susceptible to diseases, and they're not agronomically the best. So how we can make them agronomically better for us, right? More adapted. Looking at more alternative products for this, right? And create consumer awareness and acceptance of this. And again, when it comes to acceptance, believe me, once you try them, it's like you're in. I just didn't have enough popping beans today to bring for 300 people here, right? But yeah. Luckily, we have two varieties that have been just released last year. They came from Oregon State University. Jim Myers is the breeder there. And he came up with these two varieties known as Poporito and Noche Nuña, okay? And we actually got to test them this year in North Dakota, 2 locations. And that's the picture that you have there. You can see them. We got hail in that trial, so those plots got a little bit affected by the hail. That's why they're kind of small and squat. I've seen them in other places, and they tend to be a little bit larger plants as well. But they grow, and they produce beans here in North Dakota. So now we have adapted material that we can use commercially to produce popping beans in North Dakota or anywhere else in the US, pretty much. So several years ago, I tried material from Colorado and from Wisconsin, and they're extremely late for our conditions. You know, it was like end of the season, everything is dry and ready to harvest, and the Colorado lines are still green and happy. It's like it's not going to work, right? These guys may work for us. Yields are okay, but I think, again, I want to try them again next year. I think it was just bad luck because the 2 locations where we had them had hail this year. So we're going to try again, and then I'll give you the numbers, the better numbers next time, okay, for yield. And again, in David's lab, they are trying to do a lot of things, trying to figure out what else we can do with these popping beans, right, in terms of products. So, you know, he's working with protein, trying to create, say, snacks, cookies, et cetera, recipes that we can use for this. So, again, if you want to talk about that area, I'm just a dumb breeder. I don't know anything, but David is the person to talk to. But yeah, something as simple as just putting these things in the microwave for 2 minutes, right, David? Is 1 or 2 minutes? Depends on the microwave, he says. Okay. Let's say between 1 and 2 minutes, you got to try, right? Seed moisture is really important. If you have really low seed moisture, they sometimes don't pop because they need that steam to be able to pop, right? But it's very simple. You can just use a microwave or something like that, or a pan. We have found that there are differences in the same variety grown at different locations may have different popping ability, right? Some of them may pop more than others. And again, I think it goes back to that idea of the seed moisture at harvest that we were talking about. But that's what that graph is showing, that the same line or the same variety sometimes may or may not have the same popping abilities, okay? If you're really crazy about trying these beans, just let me know and I can, or just stop by our NDSU lab and we're happy to provide you some and give you a tasting, okay? So we're gonna continue working on this. I'm gonna continue giving you updates. I know there's several companies that are really interested into this and want to jump into this opportunity. I mean, there's so many opportunities here for increased bean consumption in a different way, right? Now we're not just thinking about the usual pot of beans or can of beans. These are beans that are ready in 2 minutes. That's a game changer, in my opinion, right there. Again, David is your guy. If you want to talk about breeding, it's me. But if you want to talk about anything else, it's going to be David about that. So last year we had a really good opportunity to collaborate with the Grand Farm and come up with an interesting trial that will kind of tackle two things: tile drainage, which is something important in our region, and zinc fertilization, right? And many of you might be thinking, okay, why you're talking about these two things together? And they actually are highly related, because what happens is tile helps you to have more available zinc in your ground, because waterlogging or flooding or high water table sometimes sequester the zinc and then it's not absorbable by the plant. So having lower water tables allows you for that zinc to be more available for you in the ground. So that's pretty much at the crop level. That's the relationship, and that's why it's important to look at those things together, right? We also have soils with high salt content and some other things. And then on the human side, it's an important thing for us, right? Zinc is an important micronutrient for us in our diets. 2 billion people in the world are affected by zinc deficiency, at this point in time, 17% of the population. So it's not just how we make zinc more available for the plant, but how we ensure that that zinc is being translocated to the beans that we are going to eat. In other words, can we increase zinc content in the seed and be more nutritious for us by doing all these things at the agronomic level, right? And of course, as always, I'm not doing all this work. It's just a teamwork that happens there. There is a lot of people involved, people from the Grand Farm as well. I don't know if anybody from the Grand Farm is here, but Ann Nobriga, there is a lot of people, Andrew, Jason, et cetera, et cetera. So here it is. This is a drone picture taken by one of my students, and pretty much is the same experiment under tile conditions and non-tile conditions, right? Same experiment, just duplicated in the two areas there. We put 3 varieties and 4 zinc rates, right? And that's pretty much what you have there. And here is Craig with a special thingamajinga that he uses to measure zinc levels at the foliar level. And he's able to do that. So he was taking a lot of samplings during that trial. That's the very sophisticated sprayer that we use to apply the zinc at different rates in those plots. And then we were taking leaf samples and also seed samples at the end of the cycle and send that to the lab for analysis and zinc quantification. We haven't got that yet, so I'm gonna just show you the results that we got so far. And again, this is just 1 year. We're gonna repeat this next year as well. But this is, again, another kind of close-up flight from the drone, showing you the difference in growth between tile and versus non-tile. It's probably not very apparent, but you can see that the tile actually has a little bit more of biomass, right? A little bit more of canopy cover. And that was actually confirmed by the data that we got from those images. So we use some, they call it biomass vegetation indexes. And in this case, we're seeing that the tile trial is showing more biomass than the non-tile. In other words, bigger plants. That's what he's saying, right? That's what that data is showing us. So that, hey, we're like, okay, that's exciting. That's pretty good, right? The problem was that here, I forgot to mention, we didn't find any yield difference between tile versus non-tile, which is something that I wanted to see, right? Because then it proves the idea that having tile drainage improves your yields. We're going to repeat the experiment next year and see if that's true or not. But at this point, we cannot say that. The one that was really interesting for us was chlorophyll. We were expecting more chlorophyll again on the tile treatment versus the non-tile, but the results are backwards, okay? So we're scratching our heads. This is the beauty of science, right? Sometimes you get these results and you're trying to figure out what happened there. What we found, it was an interesting relationship between zinc, tile drainage, and yield, where sometimes if you apply high rates of zinc under tile drainage, and that's the bar that you have on the right, you're going to have better yields, pretty much. That's what it's showing you. So we were able to see a little bit of a yield effect just by that zinc application. And that was really interesting to see. Now we want to kind of dive deeper into what's going on in that case, right? It's at least really interesting for now. Also, when we take leaf measurements at different points, we found out that at least at the leaf level, the plant is absorbing more zinc. So you have your zero treatment, no zinc applied, rate number 1, rate number 2, and rate number 3, which is incremental, right? So the more zinc you apply, the more zinc is being absorbed by your plants, which is good. Now we need to make sure that that zinc goes from the leaves and the stems to the pods and the seeds, right? And hopefully it's going to create a nutritional benefit for us next year, right? To be continued. And that's what we're doing for next year. Other thing that we learned about this trial, and probably you can see it in this picture, again, your tile, it's on the right side, the non-tile is on the left. You see on the tile side, on the right side, you see tiny green dots. I don't know if you can notice. Those are weeds. So here is the thing, remember, weeds also like the tile ground, right? Because they grow better there. So there's more competition for weeds under tile drainage. So be careful of that. You have to be conscious that same as your crop likes the tiling, the weeds also like the tiling because it's better drainage, better conditions for growth. So we're gonna have to be very careful about plant competition and weeds, especially on the tiling. And maybe that's what is causing that we're not seeing the drastic differences in yield, right? All right, next topic. I don't think I need to preach to the converted here. Everybody knows that beans fix nitrogen from the atmosphere through a relationship, a symbiotic relationship with bacteria, and that bacteria, we call it rhizobium, right? So everybody here, I hope, knows that. Legumes fix nitrogen from the atmosphere, so we don't need to put as much nitrogen as we do for other crops, right? That's the beauty of legumes. They increase the nitrogen in your soil and everything by just creating that association with that bacteria that you see growing there. So Barney Gates is a faculty in the microbiology department, and he's an expert in rhizobia, right? And he came to me and told me one day, Juan, how come you're doing all this amazing breeding for the beans, but I don't see anything happen with the rhizobium? We're still using the same rhizobium that was used 40 years ago. Same strains, nothing new. How we find a better strain that is going to be more productive. And also the other issue with beans, if you see in that graph on the left side, beans are poor fixers compared to many other legumes. So I mean, that graph is showing you how much nitrogen is actually fixed using symbiotic associations in different legumes. Beans is the worst. Why is that? The reason for that is beans are promiscuous when it comes down to this relationship with the rhizobia, which means that, you know, they're like me, they get along with everybody, right? So they create these associations with many different bacteria, and a lot of those bacteria are probably not the most efficient nitrogen fixers. That's the issue, right? So how we find an elite strain of rhizobia that is competitive, but also is very good at fixing nitrogen. That's what it will be elite, right? It's able to outcompete everybody else in that soil and colonize the roots. And then after that, it's able to really be good at fixing nitrogen for those plants, right? So that's an elite strain. That's the superstar rhizobium that I'm talking about. So with Barney, we're trying to work on that. And also Divita, she's my postdoc working on that. She's here as well. She's the one doing all this work, by the way. I'm not doing this, right? So back in the days, in order to screen for different rhizobia, you could only do it in groups of 2 or 3. With new molecular techniques, you can do that. In one pod, you can put 100 strains together and then see which one is more competitive. So we took advantage of that methodology and started looking at these things, right? And pretty much it tells you, you go grow those things into pots, harvest, and then with molecular techniques, it will tell you which ones were more abundant, which ones grew more and which ones grew less. And that's what that graph is showing you there. So this is how it looks in the greenhouse pretty much when you're doing the assay. And then you harvest that, take it to the lab, and do all the molecular assays that I'm talking. At the end of the day, what you get is something like this. And this is for P, because P is a little bit more ahead of us on this work. But what you have in that graph is 100 rhizobia strains tested against 4 P varieties. And what you're looking for is for those bright spots that shows that there was a lot of activity and competition for those specific strains across multiple varieties. Those are the competitive strains, right? And then on top of that, they check which one of those are being good at fixing nitrogen. So they took that to the field with the selected one, and they were able to get 16% increases in yield just by using the correct strain in that inoculant. Look at that compared with the, I don't think I have a pointer here, I don't, no. The middle bar is the commercial inoculant. And then the purple bar is your new inoculant, and you can see the difference there. This is in the field. This is not in the lab or the greenhouse. 4 sites in the field. So we're doing the same for the beans now, right? We're a little bit behind, but not only 100, but we got to collect 300 rhizobia isolates from North Dakota and Minnesota. And we did the same thing. That's what it's showing there, right? So don't worry about this. You're just interested in the bright spots in that graph, right? So you have certain isolates that are really good for specific market classes, right? Because we test them against black kidney, pinto, other things, right? But you also have some other interesting ones that are really good across different market classes. We even put some of these under high salinity conditions to see if some of them will still be able to compete, and some of them are, right? So out of this group of 300, now we ended up with a group of 12, 14, and out of those 12, 14, now we have 5 really that we really like, okay? So again, to be continued, next year, hopefully I can come back and tell you we found the new rhizobium superstar, and a company can start producing inoculum of this, right? Okay, soybean cyst nematode. People ask me a lot about this. What you're doing about soybean cyst nematode? What things are happening? And we're always doing this. I mean, several years ago, we released a variety known as ND Falcon that is resistant to HG type 0, right? Here is the problem. We have a new HG type in-house. The population of SCN in the region have changed, right? So we need to continue working on this. And again, it's mostly these 2 students, Pravat and Dalvir in plant pathology, that are doing all this work, with the direction of Dr. Whipping Yan. But it's really interesting. Be careful. It's very easy to confuse soybean cyst nematode with the rhizobium nodules, right? So you gotta be careful about what you're looking for or what you are. So again, I think everybody knows that SCN is a bad thing for our agriculture. It's really hard to control, as many of you know. $1.5 billion lost just in soybeans. We don't even know in dry beans. We think that in the case of dry beans, we're talking about 30 to 50% losses in the worst cases, right? So you can see in that graph what happens when you have no infection, you have middle infection, and when you have a high level of infection in those plants, right? They get severely affected by that. Management strategies, I don't need to probably get too much into these. Again, host resistance is what I can do as a breeder, so that's my goal, right? And again, we were able to do this for HG type 0 several years ago. Guess what? Plant pathology group came, and so they conducted surveys about 10 years ago, 2015 and 17, and then they conducted the survey again in 2024. And that's the gray and the blue bar that you see there, right? And then the different HG types that we use. Think about them as races or strains of the same pathogen, right? Look at what happened. Earlier, HG type 0 was the predominant HG type, the predominant population in North Dakota and Minnesota. Look at what happened now. It's HG 2.57, the one that is really causing trouble now. So the resistance that we had in Falcon probably is no longer effective in this case. We need to come up with something new, right? So let's go back to the drawing board. and start screening a bunch of materials. So we took about 263 genotypes, including, breeding lines from my program, cultivars that are commercial, and also germplasm accessions from that USDA collection I was telling you about, right? And the phenotyping takes about 60 days. It's painful. So again, I'm not doing this. It's mostly the students taking care of all this work here. But it's a lot of work. That's what I want to make sure that you realize, okay, that goes behind that. Then they calculate something known as the female index, and they have a scale for that, where pretty much if you get a female index of 0 to 9, your line is resistant. If it's 10 to 30, it's moderately resistant, et cetera, et cetera, et cetera. When we screen all that material, unfortunately, we didn't find anything that we could call resistant. Bad news, right? The good news is we have 29 entries that are showing at least moderately resistant symptoms or tolerance, right? So now we're taking that group and really trying to get additional crosses, studying them better, understand what the genetic factors are associated with that, et cetera, et cetera. Okay, so we continue working on SCN. That's the important message there. And there is hope, right? Not everything is close. Don't think that because you have 0 resistance, then everything is done. I think tolerance is also a good thing. Sometimes it's actually better. You put less pressure on the pathogen to overcome the resistance with tolerance versus full resistance, okay? Finally, I just want to tell you, I'll give you a big kind of general brief idea of all the tools that we're trying to use in the breeding program to make our lives more efficient. I don't know if better or easier, but at least we can probably churn more samples and look at more lines using all these new modern techniques. It takes about 7 to 10 years to develop a new variety. I don't know how many of you really know this. So it's a long work of patience and perseverance and testing that breeders need to go through, right, for most crops. Imagine being a tree breeder, right? It will be even worse, right? And an optimal variety is a combination of many things, right? And that's really what makes my job challenging. Every time someone comes and say, oh, I want this trait on my variety, it's like you're adding another layer of complexity to the whole formula, right? Because now I have to put it. The challenging is not to work in one trait. The challenging thing is to blend all those things together into what we call a variety. That's the real challenge. If you tell me, Juan, your job is just to breed for soybean cyst nematode, oh man, I'd be so successful, right? Just working on one thing. But that's the problem with the breeder, right? It has to be pulled in so many directions and so many traits. So hopefully these tools are helping us to do this. I don't know how many of you met Phil McLean. He was a faculty in our university, passed away last year after a stroke. I don't think there was a person who knew more about bean genes and bean genomics than Phil. So that was a big loss. It's going to cost us a lot of time to figure out how to fill that gap in our community. I mean, he was huge when it comes to bean genomic research. I know many of you probably don't interact with him on a direct level, but he was always behind the curtains, right? Trying to figure out all these genes and what they do and what their function is. So there is a concept that is very popular now, known as genomic selection, and it's not necessarily new in breeding. Animal breeders have been using this for a while. If you think that plant breeding is expensive, developing bean varieties is expensive, try animal breeding, okay? That's really expensive. So what they do is they have what they call a reference population. They take a bunch of measurements on those animals and also take DNA, right? And develop what they know as a prediction equation. Pretty much imagine I'm trying to forecast or predict the success of your kids based on your genomic profile, right? That's pretty much what we're doing here. Try to predict the performance of your kids based on your genomic profile, right? In simple terms. I don't know how many of you have heard about digital twins. That happens in sports a lot. They're using it to improve sports performance. The athletes, the swimmers of the Olympic team are using this. Also, they can get DNA out of you and in the computer try different medication combinations that will be specifically suited for you in that way. So there's a lot of cool stuff that is happening. We're trying to implement some of those things in a more simpler level, right? So we have, same thing, a reference population. We run a bunch of DNA markers into it, develop an equation, and try to predict what's going to happen with the progeny or of those crosses that we made, right? For that, we work with a company called Thermo Fisher to develop what we call a DNA chip. It has 4,000 markers that kind of scans all the bean genome, right? And gives you information about them. And with that, then we try to predict what's going to happen in that case, right? It costs about $17 per sample to do that, which is way cheaper than me trying to put all those lines in the field and testing them, which is expensive, right? So I'm saving money by doing this in the way that we did it for like 8 traits, including size, yield, many other factors. And as always, it works really well for some traits, but for some others, we're not getting like really good accuracy. We're still working on it. So for example, we don't get good prediction for yield. Sorry, we need to keep working on it. It's okay, but it's at the 30%. I'm not confident that that's going to help me. Moderate accuracy for diseases like common bacterial blight. And we have really good results when it comes down to seed size, for example. I'm just going to show you quickly one of the examples. This is for common bacterial blight. So that graph shows you in the x-axis, you have what we score visually. And then on the y-axis, you see what the model is predicting as being resistant or susceptible, right? And so each one of those dots is a breeding line, let's say, from the program, right? And if we think about it, we use a visual score between 1 to 9, right? Where 1 is really resistant and 9 is really bad. So in a way, we could take all those red and orange spots and think, okay, all this is susceptible. These guys are going to be susceptible to bacterial blight if I put them in the field. So why not we just erase them, eliminate them from the program, and not even think about putting them in the field, right? That's what we call in breeding negative selection. So you're getting rid of the trash and still not sure about what's going to happen with the rest. But now you know that you're eliminating the stuff that you don't need to test in the field. So it helps us to be more efficient by simply getting rid of the trash. You get it? You see it? Okay. Just trying to make sure that I'm not getting like too technical here and we don't know what's going on. In the field, we're also doing a lot of things on what we know as high throughput phenotyping. We're estimating days to maturity just flying drones, for example. And again, my student Maria is here and she's the one in charge of all this amazing work. We're also using these little carts to walk through the plots and take pictures and try to estimate yield based on those images, right? All this is done again with other people, and Paulo Flores is probably the main mastermind here of all this work. He's at the Agriculture and Biosystems Engineering Department. So they are the ones that really work into the Precision Ag stuff, really great collaborators. They developed this cart that has those depth cameras. So when they push that cart through the rows, it's taking pictures of the plants and the images. And then they're able to develop algorithms that are able to detect the pods. And then we can count pods. And as we know, pods means yield, right? So what if we can use the images to forecast or predict yield in many cases, right? And in my case, that I have to harvest thousands of plots, I don't need to harvest all of them. I just need to harvest the ones that the model is telling me that they are pretty good about it, right? So again, I don't want to get into the details here, but this is pretty much the pod counting algorithm. If you want to know more about it, just ask me and I'm happy to tell you more. My student Maria is here. She can tell you, she's the real expert. She's also working in the greenhouse with this system that allows you to take images of plants and seedlings in the greenhouse, right? I know some of the guys were able to visit the greenhouses this week. The group from Farmer Bean, I think, was there, and they were really impressed about all these things. So we're using this to scan images of plants that we have inoculated with specific diseases. And then instead of us going one by one, we're talking about thousands of plants that we have to score visually. You just use the camera, take the images, and the algorithm is going to tell you which ones are resistant and which ones are susceptible, right? Much more faster. We can probably increase the number of plants that we can screen. So we're doing it right now for bean rust. It looks something like this. I don't know if any of you have seen bean rust before, but it can be pretty bad. But then when you're scanning through, you can find things really interesting. Look at the image on the right side. You have things that are extremely susceptible and things that are very resistant, right? And these cameras are able to detect that difference right there. So you got to think about all these as I always tell people, you know, these are just tools. We're just using all these tools that help us to be better at what we're doing. Some of them more expensive than others, but I think there's worth at least trying and then see what happens in many cases, right? I think that's all I have to say. Just a little shout out for an important event that is coming in February 17th. It's a webinar called Getting It Right. You have the list of speakers there. They're gonna be talking about different things like weeds, white mold, which is really important. I didn't cover our work in white mold today. Barney's gonna be talking about the rhizobium stuff that I was telling you about. My assistant, I'll be traveling that day, so I won't be able to be there, but my assistant is gonna be presenting. You have entomology, markets, et cetera, et cetera. Really worth trying that seminar, that webinar. There is another one about nutrition that is going to happen probably in April or May. So keep your ears tuned. With that, gosh, I have 3 minutes left. I did pretty good. Really? Okay. Oh yeah, because this is 50. Yeah. So this is amazing. Yeah. Thank you everybody for taking the time to listen to all the things that we're doing. Thanks to North Harvest Beans for all the support. I mean, and this is not a support of you're doing a good job, keep going. They actually put money into this, right? So they are big supporters of the program, and they get the benefits of the varieties as well through the checkoff system. So that's really important for us. But you know, we don't want to rely only on the growers and the farmers to do all this work. That's why we get all these federal grants from USDA and other places to be able to do all the things that you're seeing here, okay? Takes a lot of people, right? Colleagues, it's not just me. I'm just lucky to stand up here and tell you the story about all the things that we're doing. But imagine all the people that are behind all this work as well. I'm gonna leave it there. My email is there. I'm gonna stay for the rest of the day if you have specific questions or interactions. And again, thank you for another Bean Day. I think I've been doing this since 2007. Okay, I'm getting old now. Thank you.